and thank you for sharing this particular part of the day, which is always very difficult with us. It is my pleasure to introduce Dr. James Powell, who is without a doubt one of the country's leading authorities in human research. First of all, let me give you a little information about Dr. Powell. He's a native Alabaman. He was born in Birmingham. He graduated Virginia Union University and followed that up with Cornell University Medical College. He's a trained clinical pharmacologist, and he trained at Cornell for that part of his professional training. And he was on the faculty there for an extended period of time before joining Procter & Gamble. And at Procter & Gamble, he was the chief researcher for that global institution. He's a former member of the Schulman IRB, the HHS Secretary's Advisory Committee on Human Subject Protection. He has been a principal investigator on many projects, and he is also the principal investigator of the National Medical Association's Project Impact. And he is the chief medical officer and co-founder of NoRx Incorporated. I think if we had a program that lasted 4 hours, James could keep us well occupied with his knowledge base. So with that, Dr. Powell.
Okay, thank you, Elbert. And John, we got to get the screen share there. I'll do that. Let's see. Okay. All right, thank you, Elbert. All right, let's see. Okay, here we go. Yeah, I appreciate the introduction, the fine introduction. I appreciate the opportunity to share a bit of my experience. Now, I was global head for the clinical pharmacology and pharmacokinetics for Procter & Pharmaceutical. Though I might have tried, I was never global head of all of the research. So just one thing I mentioned there. But I've had a long interest in the issue of clinical trials and clinical research relative to health disparities. So I've had an opportunity to do presentations to a lot of different groups, certainly community groups, but certainly I've trained probably close to about 500 healthcare providers on what it takes to be a clinical researcher. And so, but I've not really done very much here. So this is a great opportunity for me to do here in Cincinnati. And I think, Elbert, you're going to have to keep me from nerding out, because this is an area that I'm very, very excited and interested about, and I want to share the information. In fact, I've given talks to community groups across the country, as I said, even in the barber chair. And I got up from the chair with very little hair after that time, but I'm going to do my best. This is my first time doing it as a Zoom. And so I'm going to do my best to inform you of how medicines are developing, where your medicines come from. So with that, let's get started. My objectives are to make sure you learn about how the process of how a chemical becomes a drug, and touch on a little bit on the regulation of those drugs, how those regulations came into being, so you understand the intent of the regulation, as well as talk about recruiting diverse population. And that's especially important. And one of the reasons we're trying to do this right now is because, as I said, most people don't know where their medicines come from. And this is particularly relevant right now while we're trying to understand treatments for the coronavirus. And at the very end, I'm going to talk a bit about investigative responsibilities and physicians as investigators, and talk a little bit about, it's always fun to talk about, not fun, but some of the things people do and try to get away with respect to investigative misconduct. It's not funny because sometimes it does cause problems, and I'll share those with you when we get to that point. But let's go to my next slide. Let's make sure that it works. Okay, what did I do? Okay, all right. Okay, that's cool. All right, here's some terms that I'm going to use, and right now I'm not going to go through a definition of all these terms, but I'm going to define these things in context except I'll mention the one that hopefully every one of you know, and I'll apologize to those that know all of these things and that some of this stuff may be redundant, but just want to make sure everybody's on the same wavelength. And that is with respect to what a clinical trial is. It's a systematic test of a behavioral or other kind of intervention in the humans to determine its effect. And in relation to the conduct of clinical trials, these are all of the things that are all the terms that I will use relative to how clinical trials and clinical development are done for new medications. Now this chart, I'm going to spend a little time on this one because this really outlines the drug development process from its beginning to actual drug becoming available for physicians to prescribe for treatment. You see on the left, starting there, we're talking about basic research where people may, where the scientists may develop a target that they go after to be able to determine whether or not there's a particular impact on the target might be useful in the treatment of disease. And once they identify that, a lot of things, what happens is the scientists in the laboratory will spend time screening different structures for their ability to impact certain targets. Now, we've gotten better at this over the years because we've focused a lot of our attention on understanding of genomics and having better understanding of the structure of cellular substructures and receptors that allows us to be very specific with respect to drug discovery and creating chemicals that interact with those enzymatic processes and receptors. But when you've got structures that you think will be of value to the treatment of disease, they go through a preclinical phase. And that preclinical phase involves looking at these chemicals in animal species. Right now, there's a great deal of attention addressed to substituting things other than animals, but still there needs to be some evaluation in a couple of species of animals to determine whether or not that particular drug has an effect and how well it's tolerated in the animal and what particular organ systems might be affected when that chemical is introduced to a biological system. Generally, what's required to get from there into humans is you have to show safety and any evidence of effect and what particular organ systems are impacted in at least 2 to 3 species, different species of animal before you can put together a package which is called an IND, Investigational New Drug Exemption, meaning they're exempted from all the regulations with respect to marketing and just for the case of its evaluation in human beings. The process requires that you submit it to the FDA, and they have 30 days to review that information from all the animals, from all the chemistry that you've done with that particular chemical. And at the end of 30 days, whether or not the FDA has said anything or not, you are allowed to initiate your clinical protocol in human beings. That protocol, you submit that to an investigator site, which would be a physician or someone who's running the trial, and it will include all the information that has been developed in the preclinical stage. And usually in a phase 1 study, they are normal, healthy volunteers, and you start at a very low dose, several orders of magnitude of the lowest effect that you saw in the animal species. So you must take cautions, obviously, from making that progression from animals to human. And in fact, my responsibility in pharmaceutical, I had the organization that identified the appropriate doses and introduced new chemical entities into human beings. And in those studies, usually, as I said, they're healthy volunteers, but in some cases, you're dealing with drugs that are toxic, such as drugs for the treatment of cancer. And in those cases, you may not use healthy volunteers. You'll use people that actually have the disease. And so once you've done, you do a trial in phase 1, the safety and tolerability, also pharmacokinetics. You're looking for the drug, what kind of effect it has in people, how well it's tolerated. You increase the dose to see what the first toxicity or what first intolerance that you might see. For instance, nausea or headache or GI distress or things like that, and then you stop. And that way you find the levels that are tolerable in humans. From there, you move to patients, and that's where you get people with actual disease. Usually, you look for a surrogate marker, such as for diabetes. You may look and see whether or not the drug lowers blood glucose. And in a number of patients, you determine whether or not, first of all, now you're looking at whether or not the drug has the kind of effect that you want it to have in people. You do additional phase 2 studies to identify what the dose range is that provides some level of benefit without evidence of any kind of toxicity or minimizing any evidence of any toxicity in a small number of people. You see there the numbers 20 to 100 for phase 1, probably 100 to 500 patients in a phase 2 study. Once you get some level of dose, some level of efficacy, what's not pictured on this slide is what's called an end of phase 2 meeting, where you go to the FDA and say, I have a drug, a chemical that I think can be a drug for the treatment of diabetes. And this is the protocol that I want to use for studying more patients to understand its safety in the treatment of patients with actual diabetes. They rarely accept your design of the protocol, I can tell you that. And so there's always a discussion or debate as to how you're going to prove that the product is safe and effective, and what kind of patients, whether the criteria for the patients that will be entered into phase 3. Now, the interesting thing about it is that you have to do what is called at least two pivotal trials. Interesting thing about it, I wondered how that came to be. And in fact, it came to be because the legislators said, put an S on the end of the word trial. And in the legislator, it says trials. So that has been determined for the next 60 years that you got to do at least 2 well-controlled, randomized, usually double-blind randomized trials in patients to get the product approved for use. And you see there that it can be as many as 300 to 3,000 patients. But also you will note that in some of the things you've probably seen in the paper or elsewhere is that for vaccines, for instance, you're going to have to do 30,000 or 60,000 people, because what you're doing is usually what you're doing is giving a vaccination and allowing the patient to continue on with their lives under protocol and determine the rate at which those patients might actually get the particular illness, such as in the case with the coronavirus would be COVID-19. So after you've done phase 3 and you put all of that information into a package, and for a new drug or chemical entity, that would be called a new drug application. It goes to the FDA for them to review and approve. All the information, all the observations that you've seen in human beings in your trials, as well as any other exposure for trials that might have been done outside the US, becomes a part of that package for them to review. And that includes everything they know about any kind of adverse events or intolerability that was observed in those clinical trials. And that is key, because that information, the dose and all of that will go into the package and eventually describe what you say in the label, what you can or cannot say with respect to its effectiveness and with respect to its safety, and which the relative occurrence of certain adverse or safety events. Now, another thing that's not in here is that it may be, in order to review that, it sometimes takes a couple of years, and the other times the amount of time it takes to review that could be 2, 3, 4, 5 years. What's not on here is what's called, a term you might hear from time to time, PDUFA, and that stands for Prescription Drug User Fee Act. And what it is in the last, I don't know, I guess probably 20 or so years, the industry has decided to pay for the review rather than the government having to pay for it. And therefore, adequate number of resources can be put towards the actual review of these drugs. Now, some people are concerned because they say they're paying to get your product approved. No, you pay whether it gets approved or not, okay? When you file it, you pay. And if they say you didn't, it's inadequate information that doesn't prove it, then you just lost the money. So you need to be aware of that. So once they look at that and approve your drugs, then of course you're free to market it. Now, a couple of things down here. I saw good clinical practice. You see good clinical practices, good laboratory practices talks about what people do, who can do it, and how they do it, and how they record what they do for the preclinical work that they do. Good clinical practices are the rules and regulations that oversee how you actually run the clinical trials, all the rules, regulations, everything, ethics that relate to how you actually conduct your clinical trials in humans. And good manufacturing refers to all the regulations and guidance and that information that shows how the product is manufactured throughout the process and eventually manufactured for the market. And all that information must be collected and maintained and stored in detail and very, very highly regulated, very, very highly restricted as to what and how you can do the manufacturing process for a new drug. So I've been involved in conversations with community groups, and they ask who's watching. A lot of people are watching to make sure this is done properly and safely when these drugs are made. Post-marketing surveillance, sometimes that may be in additional clinical trials for follow-up, again, looking for things that probably weren't in the package that the FDA may say, you need to do more work to be able to understand certain elements that were not in your NDA package. Unfortunately, quite often, one of the things that would be in that package is insufficient levels of representation of minorities in those trials that you use for approval, and they may ask you to do a post-marketing surveillance study. And I'm going to talk a little bit more about that later because that can be an issue. Now, I'm just putting this in to let you know why drugs cost so much. The process I just shared with you in getting a drug to market can be $1 to $2 billion for one drug. You get started with a number of compounds, you might get 5,000 in discovery, 20,000 in discovery. You get 250, or maybe a few hundred that get to preclinical research because it's safe enough and well tolerated enough in animals. Based on that, you may get 5 that you want to invest in that goes into actual humans. And if you're lucky, if one makes it out of that process. The interesting thing about it is if you get to phase 2 or phase 3, and you've just spent $800 million, which I was on a project where that actually happened, and you find an adverse event that really will not allow you to market a product. And that's $800 million that's gone and nothing you can do about it. And so a lot of times, there are a lot of iterations through this process to find a product that actually makes it a market and actually useful for human beings for you to be able to prescribe it. Now, what these clinical trials I spoke about, in the law, they identify what is called a learned intermediary. Most of the times that's a physician, but some clinical trials, of course, might be conducted by dentists, and I expect some are done by nurse practitioner. But for the most part, we're talking about clinical trials that are conducted by physicians. The fact of the matter is that less than 5% of physicians choose to be an investigator. And for whatever reason, the amount of effort or the amount of support that's necessary to do the work, 70% of first-time investigators don't become second-time investigators. So that's a number that hasn't changed very much, but I think it's going to change very soon because I think technology is changing that will allow investigators a little bit more infrastructure support to be able to participate, physicians to be able to participate in trials. 80% fail to recruit as contracted, and 20% fail to enroll a single patient. And the industry invests a lot in developing those capabilities for those particular sites, but a lot of time they're dry holes and you don't get any return for the investment to get investors involved, investigators involved. But the one thing, and this is my anecdotal experience from going across the country and talk with investigators, 100%, and nothing is 100% these days, but I can tell you that every time I talk to a physician, every one of them says that their experience in being an investigator in clinical trials is allows them to provide better care to their patients because of better understanding of the things that need to be done to really monitor clinical trials and understand how disease work and how drugs work. It leads them to provide better care to their patients. One of these days I might do a study on that, but I haven't done that yet. Now, I'm not going to go through this, but I am going to show you this because, and again, this is not meant to discourage people from being investigators because I want to encourage people to do it, but I want people to know what they're looking at. And this is basically an abbreviation of the contract between the physician investigator and the federal government for those investigators who take part in those trials. It says, I agree to a lot of things, and they hold you to it. And this is, like I said, a contract. And the important part is you can, at one point I always tell people, you can delegate authority, but you can never delegate responsibility. You can't say the nurse did it or somebody else did it. If it happened, you're responsible. Was there coming? Okay. I'm gonna move on because this also is very interesting for the nerd in clinical research. And let's talk about the regulatory progression because I find it quite, and some of these things you might know, I find it quite interesting. Just about every change or every major change in regulation for drug development has happened because of a catastrophe. If you look at probably the very first one, we're talking about the Pure Food and Drug Act, and some of you have heard of the Upton Sinclair, The Jungle, the book that was written back in 1906, and talking about, actually it's talking about the meatpacking industry and the lack of what people labeled on drugs as well as foods had probably bore little results to what was actually in the package. And I tell, sometimes they called it, back then they called the legislation against quackery. And the interesting thing about it is they talked about, for instance, drugs that were used, there's advertised that can treat tuberculosis, rheumatism, everything. And what it turns out to, and cancer as well, and what it turned out to be, probably most of them was 90% alcohol. It wasn't that it was curing the disease, it's just that people didn't care after they drank it, probably. But anyway, and moving on to 1938, this is one of the more larger tragedies in the clinical research enterprise, and that is the marketing of sulfanilamide. And some of you may know about that, and that this product was put into ethylene glycol because it was a sweet mixture and it was used to treat kids. And the unfortunate thing about it is gradually they begin to see kids dying. And actually 107 kids died as a result of exposure to ethylene glycol and sulfanilamide. And the 108th person to die was the pharmacist who created it, who took his own life as a result of what he observed. Now, in 1962, I'm sure most of you have heard of that, the Kefauver-Harris amendments, where you now already have, the drug has to be pure and labeled for what's in it, and the drug has to be safe. That's from 1938. What happened in 1932 is we had to have proof that the drug was actually effective. Before then, you didn't have to prove it was effective for what you advertised it for. And that came about as a result of thalidomide, which a lot of us already know about, that the drug that was, and I'm sure our moderator knows about it because in a matter of fact, it was given to women who were pregnant and who had the deformed kids as a result of their exposure to thalidomide. The interesting thing about thalidomide, it has found a new life in the treatment of Hansen's disease or leprosy. In 1979, very interesting, the Belmont report came about. And a couple of things to talk about there, and that is, and I'm going to talk about it a little bit more, but it came as a result of observations and occurrences that occurred at Willowbrook School for the Mentally Retarded, where kids, interestingly, the people got hepatitis anyway as a result of their being housed in that facility and exchanging bodily fluids as a result of their illness. But then they tried to, they did some experiments to actually give the kids, the people, I shouldn't say kids, they're all ages that were given hepatitis and studied. And that was found to be obviously unethical and inappropriate. And the gentleman who did those studies, I think he may even still be alive and still defends his actions. But also the thing that led to Belmont report was the Tuskegee, and I'll spend a bit more time talking about the Belmont report later. You know about NIH Revitalization Act in 1993, where it's actually NIH, but it came about as a result of the AIDS epidemic. Women wanted to be a part of the clinical trials that were conducted to be able to demonstrate the safety and effectiveness of medication for treating AIDS. And that was one of the primary movers for the NIH Revitalization Act in 1993. It said in NIH trials, you had to have women and minorities in clinical trials. It does bear no relationship to whether or not minorities are required for drug development trials under the auspices of the FDA. And in 1999, the issue of conflict of interest reached headlines because of Jesse Gelsinger, a young man who had a disease called a deficiency of ornithine transcarbamylase. They had a new treatment. They put it in, the treatment was a gene therapy. They put the enzyme in a adenovirus vector and gave it to this young boy, and he had a severe reaction to it, and in fact, it took his life. Learned later that the investigators and people who run the trials actually had an investment in the company and had some other reasons that they should not have been involved in that trial and should not have involved Mr. Gelsinger. Most recently has been the FDA Safety and Innovation Act. And there has not been a specific tragedy there, except the lack of representation and some of the things that I think that tragedy has happened, but just have not been observed. And we can talk about that in a few minutes. Now, why should we be concerned about clinical trial diversity and level of representation? We know that there are a lot of things that influence how a drug behaves and how a drug works for people. Genetics, environment, habits, practice, lifestyle, all of these things that might vary with ethnicity can impact how well a drug is absorbed, how well it binds to receptors inside the body, how well it is metabolized and excreted. And all of those will affect ultimately how well the patient responds to the product. And therefore, I feel strongly, and others, and I work with many others who feel strongly that we must pursue ethnic balance if we understand how drugs work in different parts of our diverse population. And what I have pushed, what the National Medical Association and our Project Impact has pushed, is that the representation must be proportional to disease burden. That means if, for instance, if coronavirus is 60% of the, 40% of the people that get it are African American or people of color, then 40% of the people in the clinical trials should be people of color, and not simply just based on population in the U.S. Some of the cases we know, there are many cases we know of that the drug response varies. I mentioned primaquine because that was one of the key factors that led to this whole field of what we call pharmacogenomics. And that was when soldiers in Southeast Asia were given primaquine as a prophylactic against malaria. And the fact of the matter is a good number of the people of color got sick. It turned out to be hemolysis secondary to glucose 6-PD, I forget the dehydrogenase, and the other, and that led to them getting hemolysis and renal failure. And that created the whole, Werner Kaylau began the effort to create a whole field called pharmacogenomics. Let me move through here. Bidil proved in 2005 a drug is used to treat heart failure in African Americans. Trial that was done in African Americans, people that reported as African Americans, shown that it reduced mortality in heart failure by 40% in those particular trials. Plavix was a drug, there's an actual, what's called a prodrug, and people who don't have the enzyme to convert it have a less of an effect in its preventing second heart attacks. Now I'm going to talk a bit about Ceravent because that's somewhat of a case study. It's a drug used for treatment of asthma. And I don't want to go through all this. I'm just spend a few minutes on it, but just to say, make a few points. The drug was approved by the FDA in '94. And based on my understanding, it had a limited number of African Americans in that trial, and the FDA requires them to do what was called a SMART study and show more evidence of efficacy in African Americans. That trial was stopped by what's called a Data Safety Monitoring Board in about 2003. And Data Safety Monitoring Board, by the way, is an independent group that, unrelated to the trial, that get to review all the data unblinded to determine whether or not the product continues to be safe to put into clinical trials. In this case, they stopped the trial because they say there was increased morbidity and actual mortality in African-American kids who were in that trial. A lot of work, and it caused a lot of upheaval. It didn't remove the product from the market because what they finally found out was it wasn't a genetic difference, but it was the way in which the medication was taken that led to an increase. It wasn't taken, there's a long-acting beta agonist, and those patients usually, I believe, are treated with a steroid, and those that weren't treated did not, had a higher incidence of morbidity and mortality. So it led to a black spot warning, and then eventually that warning was removed. But my problem, and the reason I think this is a key issue, is what happened between 1996 and 2006 when we found out how many people were treated with that product in the African American community, and what actually was the morbidity and mortality of patients that were using that product. We don't have that information. And the reason that is important, because it's here in the next slide, what I'm showing you is what's called drug trial snapshots. These are product summaries of products that are approved by the FDA. This is as a result of what I mentioned, the FDA FDASIA that was started in 2012. It required the FDA to put a summary of demographics and other information that led to the approval of the drug and make it publicly available. You can find that at the FDA website. I have it down there at the bottom of the screen, but just go to fda.gov and put in snapshots. But what you find is that 80% of drugs approved annually have in their label up to 80%, as many as 80% in any given year. It shows that inadequate data that shows safety and effectiveness in non-white patients. And I put just one example down here of Kolanor, was approved in 2015 for heart failure, which we know occurs quite frequently and highly prevalent in the minority community. But as you see, it's not reflected in the actual data that's used for that product to be approved. So examples like things like CERAVENT perhaps could be happening, but the fact of the matter is not being studied adequately to be able to show. Especially, it may be difficult to discern for a disease like heart failure where there's an expected level of morbidity and mortality. But the concern is whether or not some of it is preventable, obviously. Okay. So I'm going to talk about why African Americans and minorities don't participate. And most of the time, people point to mistrust. I'm going to talk about that in a minute, but mistrust that has originated usually from abuse. We talk about slavery and people that were participating in experiments and even experimental surgeries who had no legal right to resist that. And the one that's most notoriously known for that is Dr. J. Marion Sims, who conducted, I think, over 35 surgeries in a woman, gynecological surgery in a woman without anesthesia. But the other aftermath of slavery includes other experiments, Jim Crow era things that were done that people participating in experiments without their knowledge or consent. The other thing, interesting, the night doctors. This is something I learned about that actually from my grandma. Don't go through the medical center at night because they'll pick you up and put you in an experiment. That's what I heard when I was growing up in Birmingham. And in the sense, people in Cincinnati, I'm sure, know about the Cincinnati radiation experiments where people are exposed without their knowledge to radiation. But the most notorious one that's always pointed to is the Tuskegee experiment, or the United States Public Health Service study of syphilis in the Negro male in Tuskegee. That's what my friends at Tuskegee make sure I say that every time. Nearly 400 men were left untreated for 40 years. Even though it was openly discussed in the medical profession and a treatment became available, they were not treated until it became news in 1972. Now, that was part of part of the thing that led to the Belmont Report. And the Belmont Report happened in '79. It was actually a hotel. Belmont was a hotel in upstate New York, where the National Commission, a National Bioethics Commission, got together and reviewed and came up with proposals for the ethical underpinning of human research in this country. These are three fundamental principles. One, respect for person. That means people must make their own choices autonomously, and that those people who are vulnerable must be protected from research. Beneficence, meaning you maximize the benefit and minimize the risk in the conduct of the trials. And justice means that the benefits of the research include the people that participate in the trials are people that share in these benefits. I think I said that right. But I'm sure our IRB specialists will let me know later. And that's Dr. Nelson. But one of the things that led to, of course, is the need for a protocol, a written instructions as to how you're actually going to conduct a trial, and the need for informed consent, the process that puts all the information and knowledge about the test agent in it so people can review it and decide whether or not they want to participate in the trial and sign. And the one thing about informed consent, it is not a contract. It is a statement that says you will participate, but you can decide, you can decline immediately after you sign it if you choose to. And of course, the need for an IRB, and that is a committee of individuals who reviews the protocol and determine all the information to determine whether it's appropriate to participate in trials. These are people unrelated to study, have no interest in the study, includes a physician, clergy, layperson, legal, and probably some others that I mentioned, but these are community people that review it and determine whether or not it's appropriate to conduct those trials. I put this slide because one of the reasons it's important that you understand all those things, because as physicians or healthcare providers, patients want to hear from you. They want to hear from those people. Those are people the people that they trust to determine whether or not they participate in a trial. And if those people, the people that they trust, the physicians that they trust, are not sharing or do not give them that information, they're not share that information with them, they're less likely to participate in those trials. And what you see is that it's the same basically for Asian, for non-Hispanic whites, for African Americans, as well as Hispanic patients. It's basically the same. So what's a physician's role in limited minority participation in trial? First of all, is people don't know about research, and therefore they don't talk about it. A lot of investigators, in fact, who do know about research, but they have certain stereotypes and biases about patients' willingness to participate, and therefore don't want to get involved in those tough discussions about things like Tuskegee and other things. These are things that we've recorded from interviews with physician investigators over the years at Project Impact. There may be some personal bias even from physicians themselves in the community, and they don't recommend their patients to participate in research. Okay, and often when physicians are asked to refer their patients to research, they're concerned about their loss of patients and therefore the loss of patients to the research facility, and therefore they don't recommend them. Why do physicians not want to participate? Of course, there are a lot of reasons. Most often they tell me they don't have enough time. Some of them talk about the negative image of the industry. That's unfortunate, but the industry does have a negative image. I think largely because people think that making money and having people pay too much money for drugs is a problem, and I agree. You may be surprised to hear me say that, but yes. But sometimes they're not supported by the employer to participate in research, and some have problems with the requirements for infrastructure to be a part of the research process. But the important point is, if you're not a part of the process, then of course, you as a potential gateway, meaning it closes out all the patients that trust you. And that's why you need to be a part of the process. And the other reason you need to understand it is because the interesting thing about it is a lot of people think you're doing research on them anyway. And so you need to understand that to make sure people understand that that is not, you're only involved in research when that information is, the information is shared with you and you have informed consent. It is illegal, unethical, and immoral and everything else for people to allow, have patients participate in research without their knowledge. And what you also see is a couple of publications that says racial and ethnic minorities agree to participate at the same rates as white when asked and when given the information. My anecdotal information is when they're given the information in a way that they can understand. So what do we do to overcome those barriers? The important part is as healthcare providers, gaining knowledge about the differences in response and why it's important for people to be a part of the process. And then we've also been very involved in advocating for diversity at every level of the biomedical research process, meaning not just as patients, but need to be investigators who enjoy the trust of patients. And I believe that every physician, regardless of race, of ethnicity, those who enjoy the trust of patients must be a part of the process. And they don't allow mistrust to be used as an excuse for poor or ineffective effort. That's an important point. We need to confront fear, mistrust, and with knowledge and understanding of research value and its protections. And we need to engage not just physicians, but the caregivers, the community, and family support in the discussion of research. People don't understand the process, and if they don't understand it, they don't trust it. And we need to develop more trusted physicians to be a part of. Okay, I've gone over my time, but I'm going to finish up here. One of the things, program we've had in National Medical Association, we call Show Me the Data. We tell, as patients and as physicians, every time someone comes to you and asks you to prescribe a medication, say, show me the data that that product is safe and effective for the treatment of my patients. And the reason we say that is because that will drive more of the pressure to develop the data that's needed to be able to understand products in a diverse community. Now, I'm going to talk a little bit about investigator misconduct. And I want to make sure I'm not scaring people, because deliberate or repeated mistakes are not what I'm talking about. Differences of opinion, honest differences of opinions, is not what we're talking about. We're talking about people who actually engaged in hiding mistakes or falsifying data. And the poster child for this, you may have heard of him, I don't know. He's Dr. Fittis. A few years ago, he was in the news. This gentleman actually had people on his staff. If he needed urine for a clinical trial, he had people on his staff just urinating, putting gallons on the refrigerator and save it for the clinical trial and just use and take it out, sort it out. And when he needed to do supply a bacteria, say from a UTI as a part of the clinical trial, he would order the bacteria from the lab and then send it in as evidence of the people having infection. So he did a bunch of things, and it paid for his BMW and his nice house in Southern California, but he went to jail. Okay, and so yeah, and the only reason, the interesting thing about it is, the unfortunate thing about it is, he got caught because somebody on his staff got upset with him and told on him. And unfortunately, that's the only way they found out he was doing these things. So that really ginned up the inspection process at FDA. But also, so one of the things that's important, that if you got somebody participating in a trial and you got falsified data in there, you're going to have to go, as a sponsor, I've got to go into that data, find that data and get it out. And if it no longer shows the product is approved, then it's likely that product might be removed, or it may be given time to do some additional work. But I'm going to end by saying that everybody has a role. Health equity for diverse populations demands a new paradigm of provider-community partnership for clinical trial inclusion. And that's an important path for us to get to precision medicine. The only question now that I have for you is, what will be your role? So with that, I'm going to ask for any questions that people might have, and let's go for it.
If I may, I would like to ask the first question, Dr. Powell. Would you comment on the fact that in spite of having an FDA-approved protocol or an FDA-approved study, could you comment as to why it is still necessary for the local IRBs to have a role in monitoring and reviewing research that is done in our areas, specifically Cincinnati?
Well, I appreciate that question, because really the IRB is supposed to reflect the local community and the mores and the ethics of the local community. A lot of times we use these central IRBs, and I'm always concerned about it because sometimes they fail to reflect some differences. For instance, as we talked about the Cincinnati radiation experience and all these other things that happen in Cincinnati may be different from things that happen in Atlanta or Chicago, New York. So we really have to reflect the local understanding and values in the community when you review and approve that program. That's why I think it's extremely important to do local RBs. Did I get it right?
I think so, but would you comment about the oversight that they provide?
Oh, yes. As we mentioned, that the the protocol goes to the IRB and is reviewed by the IRB, and they have the authority to allow or disallow the conduct of that trial in the community. And in fact, if the information about that IRB is included, I believe, in the informed consent, so that if you feel that something is not being conducted appropriately, as a patient, you have the opportunity to share that. And the IRB can in fact say, okay, we need to stop this. And we need to make changes. So the IRB really is the local, although we talk about FDA and all these other things, the IRB is a local authority as to whether or not it is safe and appropriate for that trial to be conducted in that community.
As a follow-up question, would you also comment about the preparation of the informed consent process as to the educational level that it's directed to?
Yeah, I failed to mention that earlier, and I should have. One of the important things about informed consent, as I said, it has to include information on the history and experience with a particular product in human beings. And it has to detail what it is that you're going to do in that particular study and how the patients are going to be studying, what their rights and rights are within that clinical trial. And it needs to be written at an elementary school level. Usually people say 4th grade or 6th grade level, so that anybody can understand it. One of the things, one of the concerns is a lot of things are getting to be too long, but in fact it's designed, it should be designed and written for the general population to be able to understand. It must not use any language that is not understood at the, as I said, in the elementary school level. I probably left something out, but any other questions?
Well, I have one final question, I think. You mentioned something in your talk about adverse events. Mm-hmm. Could you explain exactly what that is relative to the layman?
Okay. Well, people call it, I call it adverse events, we call it, people call it side effects, an unexpected effect that occurs after associated with exposure to a medication. An adverse event or side effect can be good, sometimes can be, most of the time we think of them as something as we said, adverse, such as a rash, or you're being treated for a headache, but you get a rash, or you get some other kind of effect, nausea, vomiting, that may occur as a result of exposure to that particular product. And yeah, basically that's what we mean when we're talking about adverse events. And as I mentioned, that the rate at which those might occur in a patient, we look at the label to see how often they might occur in patients, and that's in the clinical trials, and that becomes a part of the label to see how frequently that might occur. But yeah, adverse events are those observations that we see in the clinical trials, and as well as in practice in actual use, as to how negative effects that might be associated with the use of that particular product.
I don't see any questions posted in the Q&A, and none in the chat area as well.
Okay. Well, if we have no other questions, then I really appreciate and enjoy the opportunity of sharing with everyone the drug development process and the values and the protections in the process, and why it is so important for us to be a part of it, to make sure that we have clinically meaningful information in diverse populations for all the products, including vaccines, new drugs, and some of the things that are currently being developed for the treatment of COVID-19. We cannot sit on the sidelines. We can't wait and say we're going to wait until the product gets approved, because it can be approved without understanding of how it works for people like you. So, or your patient. So I really thank you for participating in this discussion, and hopefully I'll get the opportunity to talk to you more, and hopefully you will attend Thursday's session with Dr. Fichtenbach, where he will talk specifically about the vaccine for the coronavirus. So with that, I guess we'll close.
Well, not quite so fast.
Oh, okay.
I would like to just say that I would like to support all of the work that you are doing in this area, and for the Cincinnati community in general. I think it is important for them to know that there are people who are working at all of our institutions, hospitals, who are heavily involved in making sure that all of the research, trials, drugs, devices are reviewed in the greatest of detail to make sure that they cause no harm. So you have a strong advocate. I know at the Christ Hospital, multiple advocates who are fully engaged in protecting all human research at this hospital. And I'm sure all the others are the same as well.
That's great. And I also got to give credit to Closing the Gap, Renee Mahaffey-Harris, for their sponsorship of this session, and their efforts to also to help with the effort to educate patients about clinical trials and why they're important and what their protections are in the process. One thing I always tell them, I've gotten in the habit of saying now, is all hands on deck. Everybody's got to get in the game now, because we're dealing with a virus that is that takes no prisoners, and we got to be a part of the process. So, are we ready, Elbert? We have anything else?
No. Everyone have a great evening and stay safe.
Thank you. Take care, everyone.